oligosaccharides belongs to carbohydrates. From that carbohydrates, one of the important carbohydrates are oligosaccharides. That oligosaccharides are compound sugars that yield 2 to 10 molecules of the same or different monosaccharides on the hydrolysis. Accordingly, an oligosaccharides yield 2 molecules of monosaccharides on a hydrolysis is designated as the disaccharides, and the one yield 3 molecules of monosaccharides is called trisaccharides, and so on, tetrasaccharides, pentasaccharides, like up to decasaccharides. Disaccharides means they containing 2 saccharides, 2 monosaccharides, sucrose, lactose, maltose. Trisaccharides means they containing 3 sugar molecules, 3 monosaccharides, ribinose, ribinose. Tetrasaccharides, stachyose, they containing 4 sugar molecules, 4 monosaccharides. Pentasaccharides, verbicose means 5 monosaccharides are present. Up to 10 monosaccharides are present in the formation of oligosaccharides. See, from that oligosaccharides, that important saccharides are called disaccharides. Disaccharides consist of 2 molecules of monosaccharides covalently bond to each other. In disaccharides, the chemical bond that joins 2 monosaccharides units is called glycosidic bond. So now we discuss here this glycosidic bond. It formed when the hydroxyl group of one of the sugar reacts with anomeric carbon on the second sugar molecule. Anomeric carbon of second sugar molecules. It readily hydrolyzed by acid. It resists cleavage by base. So disaccharides mainly linked by the formation of glycosidic bonds. Composed of 2 monosaccharides called disaccharides, cells can make disaccharides by the joining of 2 monosaccharides by the biosynthesis. Like here it is glucose plus glucose, sucrose is formed, is also called table sugar. Glucose galactose formed by the glycosidic bond is called lactose. It is milk sugar, it present in the milk products. Glucose and glucose formed by the glycosidic bond is called maltose. It is also present in the starch. Here it is sucrose formed by the glucose fructose molecule by the glycosidic bond. Lactose formed by the one glycosidic bond, glucose and galactose. And maltose formed by the glucose molecule by the glycosidic bond. Now let's we discuss here it is how to form glycosidic linkages in the carbohydrates. See, carbohydrates bind with the glycosidic bond. The glycosidic bond present between the 2 sugar molecules. See on this board, we discuss here it is one glycosidic bond in maltose formation. In maltose formation, here it is sugar molecules. Glucose molecules are here it is first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. Here it is another glucose molecule, first, second, third, fourth, fifth, and sixth carbon atoms. Between these glucose molecules, glycosidic bond is formed. This glycosidic bond is called alpha 1,4 glycosidic bond. So how to form this alpha 1,4 glycosidic bond, we'll discuss here it is. That alpha 1,4 glycosidic bond and beta 1,4 glycosidic bond, beta 1,4 glycosidic bond in the lactose, alpha 1,2 glycosidic bond in the sucrose, alpha 1,4 glycosidic bond in the maltose. So glycosidic bonds are mainly alpha 1,2 glycosidic bond, beta 1,4 glycosidic bond, alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bonds. Alpha 1,6 glycosidic bonds. So alpha 1,6 glycosidic bonds present in the polysaccharides. So first we discussed here it is how to form glycosidic bonds in the carbohydrates. Glycosidic linkage is a bond formed between one carbohydrate with another carbohydrate. See, glycosidic linkage is commonly formed when two glucose molecules are linked. Two glucose molecules are linked in the maltose. The glycosidic bond is formed between the first carbon atom of one glucose molecule and fourth carbon atom of second glucose molecule. During this bond formation, the hydroxyl group of first carbon, is eliminated along with the hydrogen atom of fourth carbon. Hydrogen atom of the fourth carbon is eliminated as a water molecule. The remaining oxygen atom is formed glycosidic bond. Remaining oxygen atom is present between two glucose molecules and hydroxyl group, hydrogen group released or eliminated as a water molecule. Now, in this sucrose, in the formation of sucrose, see on the first carbon atom of glucose molecule, second carbon atom of fructose molecule. Between these molecule, first carbon of one glucose, second carbon of one fructose, glycosidic bond is formed. Here it is, alpha means on the glucose molecule hydroxyl group present below the ring. So below the ring hydroxyl group is involved in the formation of glycosidic bond. This bond is called alpha 1,2 glycosidic bond is in the sucrose. Beta 1,4 glycosidic bond. Beta 1,4 glycosidic bond present in the lactose. See in the lactose, galactose containing beta. Beta means hydroxyl group present above the ring. Hydroxyl group present here, it is above the ring. So it belongs to beta galactose. The first carbon of hydroxyl group present above the ring, it can bind with the fourth carbon of glucose called beta 1,4 glycosidic bond. Beta 1,4. Beta means hydroxyl group present above the ring is called beta. They present on which carbon? It is first carbon. This carbon react with or linked with another glucose molecule of fourth carbon. So this formation of glycosidic bond, here also one water molecule is eliminated. Here also one water molecule is eliminated. So in lactose, beta 1,4 glycosidic bonds are formed. In next maltose, two glucose molecules are involved here. It is the two glucose molecules belongs to alpha glucose, alpha glucose. Alpha means hydroxyl group present below the ring. Here also alpha group present below the ring. See one group, one glucose molecule, first carbon containing hydroxyl group below the ring. It can linked with the second group of glucose molecule. The second group of glucose molecule containing fourth carbon. Fourth carbon is linked with first carbon of another glucose molecule. So these both glucose molecule mean first glucose, mainly first glucose molecule belongs to alpha. So this glycosidic bond is also called alpha 1,4 glycosidic bond. Alpha means hydroxyl group present below the ring. So the first glucose molecule of first carbon linked with the fourth carbon of another glucose molecule with glycosidic bond. Here also one water molecule is eliminated. So these are the types of glycosidic bonds in the disaccharides. Sometimes in polysaccharides. See, polysaccharide like in starch, like amylopectin, amylose. See, here it is 1,4 alpha 1,4 glycosidic bonds are normally present between the two glucose molecules. Sometimes the amylopectin containing lateral chain like structure, between first glucose molecule. Here it is first carbon of one glucose molecule. It is the sixth carbon of one glucose molecule. First carbon of one glucose molecule, sixth carbon of another glucose molecule. So this formation of glycosidic bond is called alpha 1,6 glycosidic bond. Alpha, why we call alpha? Here it is the hydroxyl group present below the ring on the first carbon. So it belongs to alpha. It linked with the sixth carbon of another glucose molecule. So this type of glycosidic bonds are called alpha 1,6 glycosidic bonds. Alpha 1,6 glycosidic bonds are present in the polysaccharides, but alpha 1,2 glycosidic bonds present in the disaccharides like in the sucrose. Beta 1,4 glycosidic bonds present in the disaccharides like in the lactose. Alpha 1,4 glycosidic bonds are present in the monosaccharides like in the maltose. So these are the types of glycosidic bonds present in the carbohydrates like in disaccharides and polysaccharides. Only in polysaccharides, in side chain formation, 1,6 alpha 1,6 glycosidic bonds are formed. Alpha 1,6 glycosidic bonds are formed. See, here it is, if in side chain formation alpha 1,6 glycosidic bonds are formed, but in the formation of straight chain structure, always alpha 1,4 glycosidic bonds are present in the polysaccharides. But in the disaccharides, sometimes beta 1,4 glycosidic bonds, sometimes alpha 1,4 glycosidic bonds are present. These are the formation of glycosidic bonds in the disaccharides as well as in the polysaccharides.